Next, the authors recovered the chlamydial IFUs in McCoy cells. This means that they took the chlamydial IFUs from the initial 3 cell lines after being treated with wild-type PMPD or the PMPD null mutant and placed them onto a McCoy cells monolayer. After quantification of recovered chlamydial IFUs in McCoy cells, they graphed the following results. The x-axis lists the cells from which the IFUs were recovered from prior to redetermination in McCoy cells. The y-axis is IFU recovered from the McCoy cells. There was a 70% reduction in the number of recoverable IFUs from the endocervical and conjunctival cells that received the PMPD null mutant strain. Although there was a reduction in chlamydial IFUs, some were still able to enter cells. The chlamydial IFUs that entered those cells were able to grow, replicate, and burst out of those cells in the same burst size as the wild-type strain. This suggests that PMPD does not play a role in the growth and replication of chlamydial IFUs in cells. Rather, it further validates the crucial role of PMPD in infectivity and early attachment in endocervical and conjunctival cells as hypothesized. The conclusions from this figure suggest that the endocervical and conjunctival cells inoculated with the PMPD null mutant had significant reduction in chlamydial IFUs. Due to the intracellular nature of chlamydia, the lack of chlamydial IFUs inside the cells suggests that the wild-type PMPD is required for early host cell interaction. Authors then wanted to assess why the PMPD null mutant infection led to reduced numbers of IFUs in the human endocervical cells. To determine this, the endocervical cells, represented by blue circles on the diagram, were either inoculated with the PMPD null mutant or the wild-type strain. The cells were then infected with 1.5 times 10 to the 4th IFU, and some were reinfected 2 to 3 additional times. The unattached IFUs, drawn as pink dots on the diagram, were taken from the endocervical cells and then redetermined in McCoy cells so that they could be quantified. They graphed the following results. On the x-axis is the number of infections the endocervical cells received, and on the y-axis was the chlamydial IFU concentration in McCoy cells. To reiterate, they took the unattached chlamydial IFUs and redetermined them in McCoy cells for quantification. Like the previous graphs, the black bars represent cells that received the wild-type strains, whereas the white bars represent cells that received the PMPD null mutant. We see that the cells that received the wild-type strain after multiple reinfections had a reduction in chlamydial IFUs, whereas the cells that received the PMPD null mutant had almost a constant amount of chlamydial IFUs after each reinfection. This data suggests that when the chlamydial IFUs were inoculated onto the McCoy cells containing the wild-type strain, the infectious bodies were able to get into the cells. Therefore, we see decreasing concentrations of chlamydial IFUs outside of the cells. On the other hand, we see constant chlamydial IFUs in cells that received the null mutant strain after every reinfection because the infectious bodies, although viable, were unable to enter the cells. This suggests that PMPD is required for the attachment of chlamydial cells to host cells prior to entering. These results support the author's hypothesis that PMPD is a virulence factor. The overall conclusions of this paper demonstrated that PMPD is a virulence factor. The authors determined this by inoculating PMPD null mutant in a macaque model. To verify reduction in infectivity in humans, they inoculated the PMPD null mutant onto human endocervical and conjunctival cell lines. The results suggested that PMPD's role is most likely involved in early host cell interaction. PMPD is only one of 9 genes that encode for autotransporter proteins. The authors noted that there was not a complete loss of early host cell interactions with the PMPD null mutant in this study. This suggests that perhaps one of the other 8 autotransporters might be compensating for the loss of PMPD in the PMPD null mutant. Of the 9 PMPs, PMPA, D, and I are the most conserved amongst C. trachomatis. PMPD is 99.1% conserved, only beat out by PMPA, which is 99.9% conserved. We hypothesized that one of the other autotransporter, or AT genes, in Chlamydia trachomatis is involved in early host cell interactions. Due to the level of conservation, we wish to explore the role of PMPA in pathogenesis. To test this hypothesis, we first need to determine if PMPA is essential for chlamydial infection. To determine essentialness of PMPA, we would generate a PMPA null mutant and infect McCoy cells to test for in vitro infectivity and growth. This would be compared to McCoy cells infected with unaltered C. trachomatis strains. If the PMPA null mutant is non-essential, we would determine infection and growth differences in A2EN endocervical cells and HCJEN human conjunctival cells, as they are both epithelial cell lines and they are representative of locations that C. trachomatis infects in vivo. If PMPA hinders infectivity, a double null mutant combining both mutated PMPA and PMPD could be created and tested for infectivity. If this new combinatorial mutant loses infectivity, we know that they are compensatory for early host cell interactions and chlamydial infections. This will be compared to wild-type C. trachomatis infection. It may be possible to repeat this with PMPI, as it is the third most conserved PMP protein.